Hello everyone, this is chapter 20 on blood vessels. So as always, we have our learning objectives in the very beginning that you'll want to go back and take a quick look at. All right, we'll start walking the wall of a blood vessel. So a vessel is a hollow tube, and running down the middle is the open space, and that's referred to as the lumen. So that's the passageway through which the blood flows. We're going to see a lumen in every tube, like the digestive tube and the urinary tube. Um, eh, so you're going to see that term a lot this semester. Um, Arteries have smaller lumens than veins do. That helps them to maintain the blood pressure, and we'll talk about that in a little bit more detail in a few minutes. The thicker walls and smaller diameters are what give arterial lumens its round appearance in cross section. So if you look at the picture, the micrograph at the bottom on this slide, the bottom picture in the left is an artery. Notice that it's round. And then when we look at a vein in cross section, it looks like it's more collapsed. So blood is carried through the body via our blood vessels. We know that already. And the blood vessels form a network that starts and begins with the heart. And you and I have a closed circulatory system. Not all animals do. Some animals have an open circulatory system. They don't have vessels. They have the what they call the blood is called the hemolymph, is sort of just blasted out and all the cells are bathed in it, and then it's just picked back up. So it's just a little bit different type of system. Ours is all a closed system where everything stays in the vessels and the heart. The vessels that are closest to the heart have the largest diameter, and as they move away from the heart, the diameter gets smaller and smaller. And vessel diameter largest to smallest, beginning and ending with the heart, is going to be arteries to arterioles, capillaries and capillary beds, and then venules and veins. So both our arteries and our veins have the same three tissue layers, and those tissue layers are called tunics. So from the most interior layer after the lumen going outward, that would be the tunica intima, the tunica media, and the tunica externa. The tunica intima is also sometimes called the tunica interna, and it's composed of epithelial cells and connective tissue. It's lined with a specialized layer of simple squamous cells called the endothelium. The tunica media is the thickest layer in the arteries. It is much thicker than the media that we see in veins, and you're seeing the comparison between the two of them over on the left-hand side there. This consists of layers of smooth muscle that's supported by connective tissue, and the connective tissue is primarily elastic fibers, and these are all arranged in circular sheets around the lumen. Contraction and relaxation of the circular muscles decreases and increases the diameter of the vessel lumen. There is an inverse relationship between the lumen size or diameter and blood pressure. When it contracts, we call that vasoconstriction. It increases the blood pressure at the lumen as the lumen narrows. So as the lumen gets smaller, the blood pressure goes up. So decrease in size leads to an increase in pressure. When it is relaxed, what we call vasodilation, it increases the size of the lumen and it decreases the blood pressure. The tunica externa is sometimes called the tunica adventitia. This outermost layer of the vessel is a substantial sheath of connective tissue that's made of collagen fibers and some elastic fibers. Its main function is to provide structural support to the vessel, protect the vessel from damage, and anchor it to the surrounding tissues and organs so it's not moving around freely. In larger veins and arteries, the tunica externa sometimes has its own blood supply, and that blood supply is called the vasovasorum. This is the thickest tunic in veins, and it may be thicker than the tunica media in some of the larger arteries. So let's spend a little bit of time looking at each of the different vessels that we can find in the circulatory system. And we'll start with the arteries. We've already learned that arteries conduct blood away from the heart. They have a relatively thick wall that can withstand the high pressure of blood as it's ejected from the heart. Elastic arteries are located close to the heart. They have the thickest walls, contain the highest percentage of elastic fibers. Then we have the muscular arteries. These are medium-sized arteries that are also known as distributing arteries. They take blood to the specific organs, and they're often named according to that organ. So, like you'll have the renal artery or the splenic artery. Next in line, we have an arteriole. This is a very small artery that leads to a capillary. They have the same three tunics as the larger vessels, but the thickness of each is greatly diminished. The critical endothelial lining of the tunica intima is intact. The media is restricted to one or two smooth muscle cell layers in thickness, and the externa remains, but it's very, very thin. The muscle fibers in the arterioles are normally slightly contracted. That causes the arterioles to maintain a constant muscle tone. In this case, that's referred to as vascular tone. We saw this in the skeletal muscle when we talked about muscular tone. In reality, all blood vessels exhibit vascular tone due to the partial contraction of the smooth muscle in the vessel wall. The importance of the arterioles is that they are the primary site of both resistance and regulation of blood pressure. The precise diameter of the lumen of the arteriole at any given moment is determined by the neural and chemical controls, and vasoconstriction and vasodilation in the arterioles are the primary mechanisms for distribution of blood flow. So a capillary is a microscopic channel or vessel that supplies blood to the tissues. That process of getting blood to the tissues is called perfusion. Exchange of gases and other substances occurs between the blood in the capillaries and the surrounding cells and their tissue fluid or the interstitial fluid. The diameter of a capillary lumen ranges between 5 and 10 micrometers. The smallest are just barely wide enough for an erythrocyte to squeeze through. Flow through the capillaries is often described as microcirculation. The wall of a capillary consists of the endothelial layer surrounded by the basement membrane with occasional smooth muscle fibers. There is some variation in that wall structure. In a larger capillary, there are several endothelial cells that border each other and they may line the lumen. In a small capillary, there may be only one single layer that wraps around uh, to contract itself. So the function of all capillaries is exchange, and for them to be able to function, their walls have to be leaky, which is what's going to allow substances to pass through. There's three major types of capillaries. Each one is categorized based according to its leakiness, and so that's going to be the continuous capillaries, fenestrated capillaries, and the sinusoidal capillaries. The most common type is going to be the continuous capillary. It's found in almost all vascularized tissue. This type of capillary is characterized by complete endothelial linings with tight junctions between the endothelial cells. Although a tight junction is usually impermeable and only allows the passage of water ions, they are often incomplete in the capillaries, which leaves these little spaces or intercellular clefts that are going to allow for the exchange of water and other very small particles or molecules between the blood plasma and the interstitial fluid. So substances that can pass between the cells include metabolic products such as glucose and water and small hydrophobic molecules like gases and hormones, as well as various leukocytes. The next type of capillary is called a fenestrated capillary. It has larger openings or pores or fenestrations. In addition to those tight junctions in the endothelial lining. These make the capillary permeable to larger molecules. The number of fenestrations and their degree of permeability vary according to their location. They are common in the small intestine, which is the primary site of nutrient absorption, as well as the kidneys, which filter the blood. They are also found in the choroid plexus of the brain and many endocrine structures, including the hypothalamus, the pituitary, the pineal, and the thyroid gland. And then finally, we have the sinusoid capillary. This is the least common type of capillary. Sinusoid capillaries are usually flattened and they have extensive intracellular gaps and incomplete basement membranes, in addition to intercellular clefts and fenestrations. So they have a lot bigger holes. It kind of gives them the appearance of Swiss cheese. They have very large openings that allow for the passage of the largest molecules, including plasma proteins and even cells. Blood flow through the sinusoids is very slow, allowing more time for the exchange of gases, nutrients, and waste. Sinusoids are found in the liver and the spleen, the bone marrow, and the lymph nodes, where they carry lymph but not blood. So same idea, but with the lymph system. And many endocrine glands, including the pituitary and adrenal glands. So there's the picture. So there they are, continuous, fenestrated, and sinusoidal. The difference is basically how big are the holes that things can pass through. Because again, capillaries are all about exchange. So let's talk a little bit about metarterioles and the capillary bed. So a metarteriole is a type of vessel that has a structural characteristic of both an arteriole and a capillary. It is slightly larger than a typical capillary. The smooth muscle of the tunica media of the metarteriole is not continuous, but forms rings of smooth muscle, what we call sphincters, prior to the entrance to the capillaries. Each metarteriole arises from a terminal arteriole and branches to supply blood to the capillary bed that may consist of anywhere between 10 and 100 capillaries. The capillary bed itself is what we're going to talk about next. So the precapillary sphincters, the circular smooth muscle cells that surround the capillary at its origin with the metarteriole, tightly regulate how much blood is flowing from the metarteriole to the capillary it supplies. Normally, the precapillary sphincters are closed. When the tissue in that area that those capillaries are responsible for need oxygen and have excess waste products because of that, the precapillary sphincters are going to open, which is going to allow a greater amount of blood to flow through and exchange to occur before closing them again. In all of the precapillaries, if all of your precapillary sphincters in a capillary bed are closed, the blood will flow from the metarteriole directly into a thoroughfare channel and then over to the venous side, bypassing the capillary bed entirely, creating a vascular shunt. So a picture is worth 1,000 words here when we're describing this. On the left-hand side, you have the terminal arteriole. So that's the smallest of the arterioles. And you notice that it has a little side street that goes off to it. That's the very center that goes right through the center of that capillary bed, which is the blue, purple, and red thing, is what's called the thoroughfare channel. It just goes from one side to the other. Then you notice the branches going off to each side have the sphincters there. Those are called the precapillary sphincters. And those, they're like rubber bands, right? And so they are going to stay closed unless we need to perfuse more of the tissue. So again, capillary beds are all about exchange. That's why we're seeing it go from red to purple to blue. We're dropping off the oxygen. We are picking up the carbon dioxide. We're taking it over to the venous side. We're also dropping off nutrients and picking up waste material. We're dropping off hormones, anything like that. So food, nutrients, anything like that that needs to be dropped off at the tissues. It's all going to happen down here at this capillary bed. So that takes us over to the venous side. So once we've gone through the capillary bed, we're going to go to a venule. So this is an extremely small vein. Post-capillary venules are going to join multiple capillaries exiting from the capillary bed, and those multiple venules are going to join together to form the veins. The walls of the venules consist of endothelium, a thin middle layer with a few muscle cells and some elastic fibers, plus an outer layer of connective tissue that is basically the tunica externa. The venules as well as the capillaries are the primary site of emigration or diapedesis, where the white blood cells actually squeeze out and go into the tissues to do their job. So in blood, we talked about the idea of a monocyte leaving the blood and becoming a macrophage. And this is where they do that. This is where they're going to squeeze out and do that process. So that gets us to veins. Vein is a blood vessel that conducts blood towards the heart. They are thin-walled vessels with large irregular shaped lumens. They are low-pressure vessels. A larger vein is commonly equipped with valves to ensure unidirectional flow of blood towards the heart and prevent backflow. We saw valves in the heart, and they did the same exact thing. Remember, we had the tricuspid and the bicuspid, and we had the semilunar valves, and their job was to keep blood going one direction, not going backwards. Veins in the legs, especially, you're going to see a lot of them there. But veins with valves, the whole purpose of those valves is the same, to prevent backflow. In addition to their primary function of returning blood to the heart, veins can also be considered blood reservoir. Since the systemic veins contain approximately 64% of the blood volume at any given time. Their ability to hold this much blood is due to their high capacitance. In other words, their ability to extend or be very stretchy. And that allows them to store a high volume of blood even at low pressure. The large lumen and the relatively thin walls of veins make them far more distendable or stretchy than arteries. So if we were to play the game, where is all my blood at any given time? We could see that 84% of it would be in the systemic circulatory system, the main system, that 9% would be in pulmonary, and 7% would be in coronary circulation. And in that systemic circulation, that 84%, 64% of it would be in the veins, 13% in the arteries, and 7% in the capillaries. So that's why they say it's a blood reservoir. All right, just a quick picture here, giving you that idea of how this whole process works, this whole circuit works that we've been talking about. So blood is going to leave the heart via arteries, and then it's going to go into arterioles, and then it's going to go out to the different organs and where there are capillary beds and exchange, and then it's going to get into a venule and back into a vein and then back up to the right-hand side of the heart. And a nice little chart for comparing arteries and veins because they are different from one another. Let's talk a little bit about your blood pressure. So blood pressure is the force exerted on the wall of the blood vessel or the chamber of the heart as the blood is about to be ejected. So this is how much pressure is that blood putting on those structures. The term blood pressure without any specific descriptors typically refers to systemic arterial blood pressure, the pressure of the blood flowing through the arteries in the systemic circulatory route. Blood pressure keeps the blood flowing from an area of high pressure to an area of low. So what keeps that blood moving all the time is the next area has a lower pressure and a lower pressure, and it just keeps it flowing. So arterial blood pressure in the larger vessels consists of several distinct components: the systolic and diastolic pressures, the pulse pressure, and the mean arterial pressures. So the systolic and diastolic pressure. So blood pressure is expressed as systolic pressure over diastolic. So you've seen that plenty of times, 120 over 80, right? That is the typical example. The systolic pressure is the higher value, the one that's on top in that number, typically around 120 millimeters of mercury. It reflects the arterial pressure resulting from the ejection of the blood during the ventricular contraction or systole. The diastolic pressure is the lower value, usually around 80 millimeters of mercury, and represents the arterial pressure of the blood during the ventricular relaxation or diastole. The pulse pressure is the difference between the systolic pressure and the diastolic. That's called the pulse pressure. And then your mean arterial pressure, or your MAP, refers to the average pressure of the vessel in all of the arteries that you have, including the arterioles. Pulse. So elastic fibers in the arteries are going to help to maintain a high pressure gradient as they expand to accommodate the blood, and then they're going to recoil. That's what a pulse is. This expansion and recoiling effect is called pulse. The variables that affect the blood flow and the blood pressure. There's 5 different variables that we talk about that influence blood flow and blood pressure. One is going to be the cardiac output. How much blood is your heart putting out um, each minute? Compliance is the ability of any compartment to expand to accommodate an increased content. The volume of the blood, the viscosity or thickness of the blood, and the blood vessel length and diameter, which ultimately has to do with its resistance. So what happens to the blood pressure as we get further and further away from the heart? So we talked about blood pressure at the heart being 120/80 in a healthy adult. But that blood pressure decreases as it gets further and further away from the heart. So as it goes from the arteries into the arterioles, it decreases. And when it gets all the way down to the capillaries, it's down to a very low 35 to 17 milligrams of mercury 35 when we start into the capillary bed and 17 when we get through. So we don't want that blood pressure to be high there. It could cause a lot of damage to those very delicate little capillaries. And it would cause you to push too much plasma into the surrounding tissues and lead to edema and all that sort of thing. But it also allows for time for exchange. So the blood pressure has gone all the way down from 120/80 down to almost nothing at the capillaries. It's very small. So now we've got the blood ready to go back through the venous side, and it's at no pressure. So we don't have a pump like we did up at the heart to increase the pressure of that blood. So now we've got blood that's going into the venules that's very low in the capillary. So it starts at about 15, and by the time it gets all the way up to the vena cava, it's at 0. So blood that's in the venous system has to be pulled toward the heart. There's 2 main factors that are going to help maintain a pressure gradient that's going to allow for that blood to be pulled back to the heart. The pressure in the aorta during diastole is very low, often approaching 0 when the atria are relaxed, and that's going to pull on that blood that's in the venous side. And then there are 2 pumps that increase the pressure in the venous system. The use of the term pump implies a physical device that speeds up the flow. So one of those is the skeletal muscle pump. So if you've looked at the model of a skeletal muscle, let's look at the leg. What you'll notice is that the vessels are threaded through all of the muscles. So they're not just they're in the places that they are because they need to be in those particular places. And so as those muscles move, they actually squeeze those vessels and squeeze the blood up. Right. You also have valves. And so they're showing you pictures of valves there. So valves work the same way as they do in the heart. We've talked about that already. So as the blood passes through the valve, then the valve closes. The valves are very similar to the semilunar valves. They look in construction, they look very similar to that. And that's going to help keep the blood from falling back down. So this is one of the reasons why you get patients up and start walking them around after surgery, if you're able to. And that helps to get the blood in their legs moving so they don't end up throwing a clot. The other adaptation that we have is called the respiratory pump. And so when we breathe, it causes pressure changes in the veins in the chest, and that helps to draw the blood in the vena cava up. So inhalation is going to drop the blood pressure in the veins of the chest, which causes the blood in the abdominal veins to move towards the chest vein, the chest area. Constriction is going to increase the pressure within a vein as it does in an artery, but in veins, the increased pressure increases the flow. Thus, the venoconstriction increases the return of blood to the heart. It's kind of like a straw, right? It's almost like moving fluid up through a straw. There are 3 homeostatic mechanisms that ensure adequate blood flow: blood direction, distribution, and ultimately perfusion. And those are going to be neuroendocrine and autoregulatory mechanisms. So the nervous system is going to play a critical role in the regulation of vascular homeostasis. The primary regulation site includes the cardiovascular center in the brain that controls both the cardiac and the vascular functions. In addition to that, more generalized neural responses from the limbic system and the autonomic system are going to have a part of that. So let's talk about the cardiovascular centers that are located in the brain. So neurological regulation of your blood pressure and the flow of that blood is going to depend on the cardiovascular centers that are located in the medulla. This cluster of neurons is going to respond to changes in the blood pressure as well as blood concentrations of oxygen, carbon dioxide, and hydrogen ions. The cardiovascular center has three separate but paired components. The cardioacceleratory system center is going to stimulate cardiac function by regulating the heart rate and the stroke volume via sympathetic stimulation from the cardiac acceleratory nerve. The cardioinhibitory centers are going to slow cardiac function by decreasing heart rate and stroke volume via parasympathetic stimulation from cranial nerve number 10 or the vagus nerve. And then vasomotor centers are going to control the vessel tone or the contraction of the smooth muscle in the tunica media, and changes in that diameter are going to affect peripheral resistance, pressure, and flow, which affects the cardiac output. A little bit about baroreceptors. So baroreceptors are specialized stretch receptors that are located within the thin areas of the blood vessel and the heart chambers, and they respond to the degree of stretch that's caused by the presence of blood. When they are stretched, they send impulses to the cardiovascular center to regulate the blood pressure, because when it's stretched, it's saying that there's more volume of blood. The vascular baroreceptors are found primarily in the sinuses, the small cavities, not the sinuses like in your head. And these are found within the aorta and the carotid arteries. So you have these specialized receptors that are reflexes, essentially. So reflexes are all about keeping you out of trouble. That when they are stretched, then they send messages to try to lower the blood pressure. I think of baroreceptors kind of like the stretchy pants you wear at Thanksgiving. You know when they're expanded. When the blood pressure increases, the baroreceptors are stretched more tightly and initiate action potentials at a higher rate. At lower blood pressure, the degree of stretch is lower and the rate of firing is slower. When the cardiovascular center in the medulla receives this input, it triggers a reflex that helps to maintain homeostasis of the blood pressure. So there's just a graph of how that would work if you'd like to see it in graphic form. Remember, the goal is always to get things back to homeostasis. You also have chemoreceptors, and they work as reflexes as well. So these are in addition to the baroreceptors. These are monitoring oxygen, carbon dioxide, and hydrogen levels, or the chemicals, and are thereby able to contribute to vascular homeostasis as well. Chemoreceptors monitor the blood. They are located in close proximity to those baroreceptors in the aorta and the carotid sinuses. They signal the cardiovascular system as well as the respiratory centers in the medulla. So the chemoreceptors respond to increases in carbon dioxide and hydrogen ion levels. So hydrogen ions are acidic. So they're going to cause the pH to go down towards 1, towards the acid end. They're going to stimulate, those types of things are going to stimulate the cardioacceleratory and the vasomotor centers. And in doing so, they're going to increase the cardiac output, and they're going to constrict the peripheral vessels. The cardioinhibitory centers are suppressed. When falling carbon dioxide and hydrogen ion levels or increasing pH occur, the cardioinhibitory centers are going to be stimulated, and the cardioacceleratory and vasomotor centers are suppressed, which decreases cardiac output and causes peripheral dilation or vasodilation, and you have homeostasis again. Another thing that is regulating blood pressure is hormones. Of course, it's hormones. Remember when I said in the hormone chapter that second semester of anatomy should just be called hormones? We're going to talk about hormones in just about every chapter. So we're going to talk about hormones today, and I'm going to kind of review the ones that we're going to be talking about to give you a second shot at remembering all of these. So the endocrine system is going to have control over the cardiovascular system. Those are going to include the catecholamines, epinephrine and norepinephrine, as well as several other hormones that interact with the kidneys in the regulation of blood volume. All right, so norepinephrine and epinephrine, these are our catecholamines. They are released, as you will remember, from the adrenal medulla, and they enhance and extend the body's sympathetic or the fight or flight response. They increase the heart rate and force a contraction while temporarily constricting blood vessels to organs that are not essential in the fight or flight response. Remember, the systems that are not essential in the fight or flight response are the digestive system, urinary system, and reproductive system. So they redirect the blood that would be going to those systems to the liver, the muscles, and the heart that can respond to an emergency situation. Okay, antidiuretic hormone, ADH, also known as vasopressin, is secreted by the cells of the hypothalamus, and it's transported via the hypothalamic hypophyseal tract to the posterior pituitary, where it is stored until it's released upon nervous stimulation. The primary trigger prompting the hypothalamus to cause a release of ADH is increasing osmolarity or saltiness of the tissue fluid, usually as a response to a significant blood loss. ADH signals its target cells in the kidney to reabsorb more water and prevents the loss of additional fluid as urine, and this will increase the overall fluid levels and help to restore the blood volume and the blood pressure. Additionally, ADH constricts the peripheral vessels. And here it is. It's back already. I told you you're going to see this like 4 or 5 times. So you might as well learn it, y'all, because I'm just not going to give up on this. You have to know about this renin-angiotensin-aldosterone mechanism. It is has a major effect on the cardiovascular system. Renin is an enzyme. Some books refer to it as a hormone. Either way, it doesn't matter. It's important to know that it's involved in the renin-angiotensin-aldosterone pathway. You have specialized cells in the kidney that are called the juxtaglomerular or JG cells that respond to a decrease in the blood pressure. So when the blood gets to the kidney, these specialized cells are going to determine if the blood pressure is too low. And if it is, it's going to cause the secrete or stimulate the secrete of renin into the blood. Renin is going to convert the plasma protein angiotensin-O-gen, which is produced by the liver, into its active form angiotensin 1. Angiotensin 1 is going to circulate in the blood and then is converted into angiotensin 2 in the lungs. That reaction, if you remember, is catalyzed by the enzyme angiotensin converting enzyme, better known as ACE. So angiotensin 2 is a powerful vasoconstrictor. It greatly increases the blood pressure. It also stimulates the release of ADH and aldosterone, a hormone that's produced by the adrenal cortex. Aldosterone increases the reabsorption of sodium into the blood by the kidneys, and since water follows salt, this increases the reabsorption of water, and that in turn increases the blood volume, which increases your blood pressure. Angiotensin 2 also stimulates the thirst center in the hypothalamus, so an individual will likely consume more fluid, and if you take in more fluid, that will help increase your blood volume and your blood pressure as well. Another hormone is erythropoietin, EPO. It's released by the kidneys when the blood flow and or the oxygen level is decreased. EPO stimulates the production of erythrocytes within the bone marrow. The erythrocytes are the major formed element in the blood, and they contribute 40% or more to the blood volume, a significant factor of viscosity, resistance, and pressure, as well as flow. In addition, EPO is a vasoconstrictor. Overproduction of EPO or excessive intake of synthetic EPO, called Procrit, often to enhance the athletic performance, will increase the viscosity and resistance and pressure and decrease the flow in addition to its contribution as a vasoconstrictor. So we talked about that in a previous chapter. That's called blood doping. Atrial natriuretic hormone. This is secreted by the cells in the atria of the heart. Some books call this atrial natriuretic peptide. Either way is fine, ANH or ANP. It's secreted when the blood volume is high enough to cause extreme stretching of the cardiac cells. So in this case, we have high blood pressure we're trying to address. So cells in the ventricle produce this hormone with a similar effect that's called B-type natriuretic hormone. Natriuretic hormones are antagonists to angiotensin II. They promote the loss of sodium and water from the kidneys. They suppress renin, aldosterone, and ADH production and release. All of these actions promote the loss of fluid from the body, so the blood volume goes down, therefore the blood pressure goes down. And then finally, we have autoregulation of perfusion. So autoregulation mechanisms require neither specialized nervous stimulation nor endocrine control. They are local self-regulatory mechanisms that allow each region of the tissue to adjust to blood flow and therefore its own perfusion. These local mechanisms include chemical signals and myogenic control, myogenic meaning muscle control in the wall of the vessel. So just a kind of a flow chart, if you like flow charts, of looking at what happens when we have a decrease in the blood pressure or volume, short term, how we manage that, long term, how we manage that. Some clinical considerations in vascular homeostasis. Hypertension, this is a chronically elevated blood pressure. It is defined as chronic and persistent measurement of 140 over 90 or above. Pressures between 120 over 80 and 140 over 90 are considered pre-hypertensive. Hypertension is always referred to as a silent disorder because you don't realize that it's happening. You have baroreceptors that we've talked about. The baroreceptors, if you don't do something about your high blood pressure, baroreceptors will start sending out warnings, right? And you don't do anything about it because you don't know about it. You don't have your blood pressure checked. Nothing happens, then those baroreceptors actually reset themselves, and the higher pressure becomes the new norm, which is why it's called the silent killer, because it just keeps going up and up and up, and you never realized you had a problem. This is why it's important to make sure that people get their blood pressure taken on a regular basis. It can lead to a heart attack or stroke. Hypertension can. It can also lead to an aneurysm, which is a ballooning of a vessel wall caused by the weakening of that wall. It can lead to peripheral artery disease, chronic kidney disease, or heart failure. There are two major types of hypertension based on cause. Primary hypertension is what most patients suffer. They don't really know what's causing it, so we can't cure it, but we can treat it. The risk factors include heredity, diet, age, obesity, and smoking. So obviously we can't do anything about your heredity, but we can reduce the salt and fat intake, ask your patient to lose a little bit of weight, increase their activity or get some more exercise, and not smoke. Secondary hypertension is only 10% of hypertension patients, and it's attributed to specific problems in the endocrine or urinary system. They know what's wrong, right? Treatment plan should focus on the case. So I am one of these people who have secondary hypertension because I only have one kidney. I was born with just one kidney. I developed hypertension in my 40s and I couldn't figure it out because I was really healthy and I didn't have any problems whatsoever with my weight and didn't have problems with my diet. And I got lots of exercise because I live on a farm and I couldn't really figure out what was going on. So I happened to have a scan because I was having some digestive issues, and that was when they realized I was missing a kidney. That's what was attributed to my hypertension because my blood pressure stays higher all the time because I only have the one kidney working. So I have to take high blood pressure medication because I only have the one kidney. Hypotension is the other end of that scale. This is when the blood pressure is chronically below 90 over 60 millimeters of mercury, and is typically not a cause of concern unless it's making your patient dizzy or uh, um, obviously passing out would be a bad thing too. So typically people who have naturally low blood pressure, it's associated with them having a longer life. But if it's something that's suddenly happened, that's a whole different issue. Another concern is hemorrhaging. So a hemorrhaging is a loss of blood that can't be controlled by hemostatic mechanisms. The loss of a significant amount of blood can lead to a condition called circulatory shock. Circulatory shock is a life-threatening condition in which the circulatory system basically collapses, so it's unable to maintain blood pressure or blood flow to adequately supply all of the oxygen and other nutrients to the tissues that they need. There's several different types of shock that I want you to know about. Hypovolemic shock is typically caused by a hemorrhage, a blood loss. It can also happen because of extensive burns, which you would lose a lot of blood, exposure to some toxins, and excessive urine loss because of diabetes insipidus or ketoacidosis. Cardiogenic shock is the result of is what happens when you don't have the heart working to maintain the proper cardiac output. So most often this happens when someone has a heart attack or a myocardial infarction, but it can be caused by arrhythmias, valve disorders, cardiac failure, or simply insufficient flow of blood through the cardiac vessels. Vascular shock occurs when the arterioles lose their normal muscle tone and dilate dramatically. And remember, the arterioles are the ones that are really maintaining the blood pressure because they're the ones that can vasoconstrict and vasodilate. And then finally, I talked about fetal circulation already in blood, but we're going to talk about, or in the heart, I'm sorry, but we're going to talk about it again, and just a reminder of the different shunts or bypasses that you have in fetal circulation. The foramen ovale, remember that was the opening or the hole between the two atria that allow blood coming that has just been oxygenized by the placenta to flow from the right atrium into the left atrium. There's a valve that's associated with this opening that prevents the blood from going back into the wrong side during fetal period. And as the newborn begins to breathe and the blood pressure in the atria rises, that shunt permanently closes. The fossa ovalis is what's left. It's the remnant of the foramen ovale, and it remains in the interatrial septum after birth, which marks the location of the former foramen ovale. The ductus arteriosus was a short muscular vessel that connect the pulmonary trunk to the aorta. Most of the blood pumped from the right ventricle into the pulmonary trunk is thereby diverted into the aorta. Only enough blood reaches the fetal lungs to maintain the developing lung tissue. Remember, there's no oxygen exchange happening out there. So when the newborn takes that first breath, pressure within the lungs drops dramatically, and both the lungs and the pulmonary vessels expand as the amount of oxygen increases. The smooth muscle in the wall of the ductus arteriosus constricts and seals off that passageway permanently. Eventually, the muscular and endothelial components of the ductus arteriosus degenerate and leave only this connective tissue band component that we call the ligamentum arteriosum. And then finally, we have the ductus venosus, which is a temporary blood vessel that branches from the umbilical vein, allowing much of the freshly oxygenated blood from the placenta, which is where all the gas exchange is happening between the mom and the fetus, to bypass the fetal liver and go directly to the fetal heart. The ductus venosus closes slowly during the first week of infancy and degenerates to become the ligamentum venosum. And then finally, just a picture of all those different bypasses that I was just talking about in fetal circulation. So this gets you ready to study your vessels and study the names of vessels. I recorded the arteries for you, and I'm going to say this again. I posted this in an announcement. If I forgot to, then it's good that I'm saying this again. When you are identifying vessels, you have to give a right or left. Most of them have a right or left. It is from the patient's perspective, not your perspective. So your right is their left, but we're always naming from the patient's side. And you have to include the term artery or vein. because you're going to see when you get to the veins that a lot of the arteries and veins have the same name. So you don't want to be confusing. So you should, to get full credit, you should have an R or an L. You don't have to write out right or left, but you can. And then the name of the vessel, and then either artery or vein. And you can use an A or a V. Either one of those is fine as well, to save a little bit of time. So if you have any questions, please let me know. See you for the next lecture.